Correct. This is the sequence. The very first point you need to mention the diaphragm. What happened to diaphragm? It is going to contract and becomes flatten, becomes flatten, moves downward. The next step is about the intercostal muscles. The external intercostal muscles contract while the internal intercostal muscles relax. I'm writing opposite just to mention. Then the last one is the volume of thoracic cavity increases, volume increases, and the pressure decrease. The important word is thoracic cavity over here. So these are the 3 points that are needed to be mentioned. Next question is, a scientist prepared an alveolar tissue to view an optical, by using an optical microscope. The scientist cut very thin slices of alveolar tissue. Why do you think he cut it into very thin slices? Exactly. They need to be thin for what? We need to give reason. Why is written? So we need to give reason for this thinning. They need to be thin as it's optical microscope. So they need to be thin so that exactly light can pass through them. What will happen if they are not thin? So exactly, it will absorb the light instead of passing through it, so the image won't be clear. So thick layer won't allow light to pass. There's no need to write this because they haven't asked about it. I'm just explaining. Next thing. So we have a structure of lungs. They have mentioned these are alveoli. Which duct is present with alveoli? Which duct, a tube or a pipe, is present with alveoli? So we have, yes, bronchiole. Yes, it's bronchiole. So we have trachea. Trachea break into bronchi. Now each lungs contain bronchioles, then therefore they divide into bronchioles, which are connected to alveoli. So this one is bronchiole. Number two, the scientist uses a ruler to measure the diameter of some alveoli. Now, this here some students get confused because first of all they need to calculate the diameter. Okay, use the information from table 2 to calculate the mean diameter in micrometer. Here, millimeter is given. How to convert micrometer into millimeter? Exactly. Multiply by 1,000. Multiply by 1,000. Okay. So first of all, we are going to take the average or the mean. How to calculate the mean? The total. So do you have calculator with you? Okay. Can you please calculate the diameter really quickly? Though I know the answer, but I want you to just calculate it really fast. 3. 3 millimeter. Now, first of all, we need to convert it into micrometer. Can you convert it into micrometer? Exactly. 3000 micrometer. Now, what we need to calculate, we need to calculate the actual the diameter. So how to calculate it? So we divide it with the magnification. Okay, magnification. Magnification. So it would be like actual size. We say that actual size is equal to image size divided by magnification. So we have 40 exactly. Yes. We already have this, the size of image. We have to tell the actual. Here we are finding the actual 75 what? Millimeter or micrometer? Micrometer. The unit should be correct. Okay. Next one. Okay. Are you comfortable in doing the calculations by, because I really appreciate if the student do the calculation. Okay, so you need to do the calculation thing. This formula is really easy. It's very simple. Magnification is equal to image size over actual size. We have just rearranged this formula. Okay, I'll ask you later what is the formula. Okay, to just check whether you are getting it or not. Next thing. Okay, if you have a scale which have 1 millimeter graduation, like 1 millimeter, then two, then three, there is no in between. There is no point 0.5. Okay, what can be the percentage error? It's very simple. What is a percentage error? Okay, if this is a scale, it's a normal scale. Normal scale has a graduation of millimeters, very small, between the centimeters. This one is centimeter. In between, they have millimeters. So, what can be the error of the scale? 1. Good. It's 1. Unit is already written. So, what would be the percentage error? Let's calculate. So, let's calculate percentage error. The error can be 1. How many total graduations are there? 4. 1 out of 4 is what? It's a percentage, right? So it's 25%. Okay, so are you good at maths? Have you opted? I can see that's why I've asked. Okay, have you opted chemistry? Okay, then we need to work more on calculations. You were right. Okay, so we need to practice a lot. Whenever there is a percentage mark, we need to put 100. Okay, shall we move to the next? So the next one says molasses is a solution that is usually we get from sugarcane. It's obtained from sugar beet here, sugar beet plant. The sugar present in molasses are sucrose, glucose, and fructose. So among these three, which one are monosaccharide? Sorry, give the number. They haven't asked about the name. They ask about how many monosaccharides are there. So there are two monosaccharides. You can also write the name, but when they have asked about the number, just give them a number. Okay. A student uses a biochemical test. Let's revise the biochemical test. Which test is used for reducing sugar? Reducing sugars. It's for proteins. Reducing sugars. It's from B. It's from B, but it's not barbiturates. Benedict's, right? So, what is the original color of Benedict's solution? Yes, the original color is blue. So when if reducing sugars are present, then this blue changes to what color? Brick red upon heating. Upon heating. Clear? So for starch, which test do we use? Iodine. Um, what is the starting color of iodine? Oh, this is the end point. It's brownish. If starch is present, then it converts to blue black. Okay. If protein is present, that is blue to violet. It's violet, and the change is from blue to violet. Only Benedict test needs heating. The other one happens without heating at room temperature. Acid? Okay. Let me. So we have fats. In fats we need ethanol, and then we add water and sorry, and then emulsion. Not emulsification, emulsion. Here we don't use the word emulsion. So another term the student usually confuse the emulsion and emulsification. So it's if we talk about the biuret one, it's basically the sodium hydroxide along with copper sulfate. That's why it's blue in color. Okay. So let's just go for this. What is the test for reducing sugar? Benedict's. Explain this test. So don't just write the heating first. First mention Benedict solution is added to a sample. Added to a sample. Here we have a sample of molasses, and then it is heated in a water bath. Oh, sorry, heat in water bath. What is the result? Hmm. The end point would be brick red precipitates, which students usually miss. You need to write brick red precipitates. Okay. Now, what is the explanation of expected result? The explanation is quite simple because it contains what? It contains two reducing sugars. What are those? Good. Glucose and fructose. Okay, we say that monosaccharides are reducing sugars. Okay, like they can directly convert into carbon dioxide and water. Simple. That's why they're reducing sugars. Sorry, what I was. It contained two reducing sugars, glucose and fructose. So the color changes from blue to brick red precipitate. Next one. So free sugar is a sugar in food and drinks released when food is crushed or when sugar is added to home or to food at home for manufacturers. Now again, a calculation is needed. It's easy. Try to understand this. So scientists usually say that 5% of the energy should be from the sugar. We should consume only 5%, not more than that. So the mean daily requirement for a 10-year-old child is 8100 kilojoules. So the free sugar in 1 tablespoon of molasses is this one. How many tablespoons should a boy a child should consume. So first of all, we need to calculate what is 5% of the total energy he takes in. 5% of total energy, that is 8100 kilojoule. So 5% mean 5 divided by 100 multiply by 8100. So 5 into 81 is, yes, okay. So this is the energy he needs from molasses. Okay. Now, if one tablespoon contains 250, how many will be there in 405? So we need to divide it. Let me rephrase it. For example, if you have three candies, nine candies, and you have to give it to three children, so you will divide it, right? So here we have a total of 405, but we need to divide it by 250. So what is the answer? And that will be around 1.62 tablespoons. Number 1.62 tablespoon. Okay, we just need to understand the question for calculation first, the very basic thing. Next one, an experiment done on this apparatus. We have molasses, we have water, we have animal bladder. It's animal bladder mean it's a semi-permeable membrane or a selective permeable membrane. Okay, so a process of osmosis is going to happen. Now what will be the movement of water to the area of low concentration? So can you tell me whether it will move in the tube or whether it will move in the beaker? There is more water in tube. Okay, okay. Just look at the labeling. Just focus on the labeling. We have a tube which contains molasses, not water. So molasses is means it's a solute. There is more solute and lesser water. So it has lesser water potential. Oh, let me explain it, then answer it. So this molasses contain lesser water potential, while this beaker is only distilled water. This will have more water potential, and moreover, we have an edge, an advantage that we have semi-permeable membrane. So water will move from high concentration, that is beaker, to the area of lesser water potential or the low water concentration. So it will move inside the cell, inside this tube. When it move inside the tube, the volume is going to be increased. So there would be lesser air present over here. Let's say lesser space for air. Lesser space mean more air pressure. Lesser space mean more air pressure. Just take an example of it. Take a balloon, press it. Lesser space mean more pressure of air. Okay. So explain why the air pressure in the tube increased. You have to tell the whole process, that how the water moves inside the cell. How would you tell this? I'll write the key points how you will tell this. Number one, you have to mention about the gradient. Gradient means you need to mention where there is more water potential and where there is lesser water potential. Mention the gradient. Number two, mention that molasses is present inside the air tube and it has lesser water potential that draws the water from the beaker into the tube through animal bladder. So we say a semi-permeable membrane is present in the form of animal bladder. So it will move inside. So the level of water in a test tube increases, the airspace decreases, due to which there is more air pressure inside the tube. Have you got the answer? Or we can also mention where I have written the airspace, there is lesser airspace. Here we can less, as well as we can say there is lesser volume if we want to use a scientific word. Mention the word volume, it will increase your marks. Next thing, so the student repeated the investigation, but one change to the molasses. Now, what does that change? The scientist, sorry, I've said student. The scientist did not change the volume of molasses at the start of investigation. The scientists observe that air pressure inside the tube increased by 160 kilopascal compared with 800 kilopascals in the first investigation. Suggests the changes the scientists made to the molasses to cause this smaller increase in the air pressure. What do you think he has done? Okay, let's rephrase this. Lesser molasses mean a dilute molasses. It would be like this. It would be dilute molasses. It needs to be like this, that lesser water should enter it. So lesser water mean there must be water present in the tube already. So it would be dilute molasses. Okay. So what is the next thing? Oh, sorry. I moved to the suggested one. Okay. Let's calculate the percentage over here. So what is the percentage? 162, 800. What is the increase? 80%. It would be 80%. Okay. So I really want you to do question number five. I will provide this figure to you if you want so. If you do you have access to papers, past papers? So when you click on it, okay, don't do this because this one is calculation. You need my help. You will need my help, definitely. Don't do this alone. Okay, I would really want you to do question number six. It's easy, quite easy, very straightforward question. Question number six, I'm giving you homework. So another homework is that you need to tell me what what things you want to discuss in paper 2 of the same year, year 24. You need to mention the questions. Please don't give all the questions because it's really tough to explain all the questions in one lesson. And I really want to focus on the points that you lag in. So try to solve paper 2 and tell me which questions you find or which question do you want me to explain. I will explain those. Okay, I'll give you the I can text you. It's okay. I'll share in the email or the chat. Second thing, I'm not giving you this paper. We can solve it tomorrow. Second thing, you need to give me a list of topics which you want to discuss in detail. Okay? I hope this lesson is useful for you. If you have any question, you can tell me now. Do you feel do you feel any improvement in your concepts or in your technique? So I'll make it easy for you. Once you practice more, like more practice means you are like more, it's more easy to understand the questions if you practice it more. Okay, so I want you to practice with me first of all, and then practice on your own. If you find any difficulty, like I've given you one paper, paper 2, go through it. Or if you want me, I can give you paper 1 of 23 if you feel so.